having discussed the transcription, I will move on into translation. Before moving on to translation, we need to understand the basics of mRNA, tRNA, and ribosomal rRNAs and all. We have discussed the formation of all these in before video. Now we will discuss the translation. Before going into the translation, we need to know the basics of tRNA. What do you mean by tRNA? tRNA is nothing but a RNA which carries the amino acids. So what are these amino acids? These amino acid sequences make up your protein. So to make up your protein, you need all these amino acids to get aligned in a particular sequence, so you get the desired protein which is required for the function. So to carry these amino acids from the amino acid pool to translation, which is occurring on the ribosomes by attachment of mRNA to the ribosomes, you need a particular RNA to carry these amino acids. This is known as tRNA, also known as transfer RNA. Now we will get into the structure and all these functions, what it can do. So going into the structure of tRNA, there are 75 to 90 nucleotides, which is the secondary structure of the protein. We know we have primary structure, secondary, tertiary, quaternary structure of the protein. Okay, but this is RNA, sorry. Yeah, secondary structure is a cloverleaf form. What do you mean by cloverleaf form? So this is what the cloverleaf form means. If you rotate this image by clockwise direction by some 15 to 30 degrees, you can get the same model like this. So this is nothing but a cloverleaf form. It have an anticodon, and what is anticodon? We'll discuss the anticodon. It has this and is at opposite 3' amino acid, and all tRNAs, both eukaryotic, prokaryotic, have CCA at the 3' end, along with high percentage of chemically modified bases. The amino acid is covalently bound to 3' end of tRNA. CCA can carry amino acids. This is a good mnemonic. So before going into T-arm, D-arm, and A-arm, all these anticodons, just let's have a human being who is standing here. Let's compare this human being with the tRNA. Okay, I'll write over here. So this is a human being structure. So this is nothing but this side, attachment side. So this is your T-arm, this is your D-arm, and here you have anticodon, and on which it is stand is mRNA, and through which stabilizes all this structure is nothing but your ribosome. Okay, I think you're cleared with the structure. So we will get into the details of the structure. So this is what they are saying is this 3' end of your tRNA has an attachment site. This attachment site, what this site does is it brings the amino acid and it attaches to the amino acid. Okay, see this process called charging. We'll discuss the charging with further on. Okay, and then it pairs to your mRNA. This is nothing but codon, anticodon. Anticodon is present on tRNA, whereas the codon is present on mRNA. How many nucleotides does a code contain? 3 nucleotides. So you have T-arm. T-arm contains T size C. This is ribothiamine, pseudouridine, and cytidine sequence necessary for tRNA ribosome binding. Okay, T-arm threatens tRNA molecule to the ribosome, which means the T-arm will carry the tRNA molecule to the ribosome, which attaches the tRNA to the ribosome. You need attachment of the tRNA to ribosome as well as your mRNA. Okay, so T-arm will attaches to ribosome, whereas your anticodon attaches to mRNA, whereas your A-arm will have amino acid. Okay, am I clear? So yeah, remember by this human structure, which should be very easy instead of this cloverleaf form model and all. I'll get into this again. See, human being, you have head, which is attachment site. Let's have a cap over him. So this is his amino acid, and this is T-arm, which have ribosome attachment, D-arm, and anticodon, which he stands is mRNA. Now D-arm contains this the part is important. T-arm contains ribothiamine, pseudouridine, and cytidine sequence. Yeah, D-arm contains dihydrouridine residues necessary for tRNA recognition by correct amino acid tRNA synthesis. D-arm allows detection of tRNA by D-arm detection of tRNA by amino acid tRNA synthesis. What is this amino acid tRNA synthesis? Nothing but which gives the amino acids to the tRNA. Okay, so this D-arm is necessary for the detection of this tRNA by amino acid tRNA synthase, which gives the attaches amino acids to the tRNA. Attachment site is nothing but C can carry amino acids. This is a mnemonic, can carry amino acids is the amino acid at the acceptor site. So basically you have 4 sites: T site, D site, A site, and anticodon site. Okay, like the 4 directions. Now we will go into the charging. What do you mean by charging? See, this is the nascent tRNA which doesn't have any amino acids, no amino acids over here. So you need to attach an amino acid. How do you attach amino acid? That process of attaching amino acids to the tRNA is nothing but charging. So how do you charge an amino acid? The charging of amino acids is done by amino acid tRNA synthase. This amino acid tRNA synthase uses ATP, one unique enzyme per respective amino acid, which means for every charging of your tRNA requires one ATP, which attaches the tRNA to the amino acid, and binding of charged tRNA to the codon is responsible for accuracy of amino acid selection. Okay, amino acid tRNA synthase matches an amino acids to the tRNA by scrutining the amino acids before and after it binds to tRNA. What does this mean? Amino acid tRNA synthesis, what does it makes is it matches an amino acid to the tRNA by scrutinizing amino acid before and after it binds to tRNA. If an incorrect amino acid is attached, see, the bond is hydrolyzed. A mischarged tRNA reads the usual codon but inserts the wrong amino acid. So what does this mean? Let's say you need an amino acid over here, amino acid 1, but misinterpreted your tRNA have brought in the amino acids 2. If it attaches these amino acids in this sequence of your protein, entire protein sequence will be disrupted and you will get another protein, and this leads to mutations. Okay, and then occurs after charging, then occurs initiation factors. We will discuss initiation factors, elongation factors in the next translation, and will leads to pairing of codon and anticodon. Okay, yeah, these are start and stop codons. Okay, yeah, this I already discussed, but we will go through this again. mRNA start codon, mRNA stop codons. What is a start codon? Start codon is nothing but for every mRNA you have AUG before starting of any sequence. See, if we go to the gym before any doing any exercise, we'll do workout. The same way, before initiating or synthesizing any protein, you need a certain codon, which is like a warm-up for your translation process. This warm-up codon is nothing but AUG. What is AUG? It forms methionine in eukaryotes. See, in eukaryotes, AU is augments the protein synthesis. Eukaryotes. Eukaryotes codes for methionine, which can be removed before translation is completed. Which it is not necessary to remove this methionine, which it might be removed and it might not be removed based on the function of the protein that is necessitated. Okay, in prokaryotes you could code for N-formyl methionine. Formyl methionine stimulates neutrophil chemotaxis. mRNA stop codons UGA, UAA, UAG recognized by release factors. What is release factors? See, for any synthesis of your protein, which is nothing but a translation, you need factors. These factors are initiation factors, elongation factors, release factors. These release factors will stop the translation, which ends up your translation. So this release factors recognizes these codons, which is UGA, UAA, UAG. How to remember this? You go away, UGA, you are away, UAA, UAG, you are gone. So these 3 codons, what they does is, once the release factors will see these codons, they'll just abruptly stop out all your translation. Okay, now we have understood the process of how is tRNA structure and how the tRNA is carrying these amino acids, what are start codons, what are stop codons. Now I will go into the process of protein synthesis. So as we discussed, you have initiation factors, elongation factors, and termination, which is by release factors. So all these factors will act in a sequential manner so that your protein gets synthesized. First, going into the initiation. And about this, yeah, in initiation you have eukaryotic initiation factors, which is EIF. Identify the 5' curve. See, you have mRNA, you have 5' and 3', and always the synthesis occurs from 5' to 3'. So this is how we written over here. Synthesis cis 5'3". 5'3". Yeah, synthesis occurs always from 5' to 3'. Elongation factors resemble 40S ribosomal subunit with initiator tRNA. What is initiator tRNA? Nothing but your methionine amino acid carrying tRNA. So once this eukaryotic initiation factors, what they does is they assemble before the entry of mRNA into the site of translation. You need all the setup needs to be set up. You have all the setup has been done by all your elongation eukaryotic initiation factors and your tRNA. So before this enter the site of translation only, all you have tRNA and you have 40S ribosome which are ready to get attached to the mRNA. So once these eukaryotic initiation factors are released, when mRNA and the ribosomal 60S RNA 60S subunit assemble with the complex required and it requires GTP. What does this mean? See, you have tRNA, you have 40S ribosomal subunit. Okay, in eukaryotic it's 80S, and whereas in prokaryotic it's 70S. We know in eukaryotic it is even number, in prokaryotic it's prime number 70S, since this occurs from N terminal to C terminal. Okay, ATP is for activation of tRNA. Already we have discussed during charging, which is done by amino acid tRNA synthase. Here GTP is for gripping and going to plant, which is translocation. What does this mean? See, once your what's the function of your initiation factors? These initiation factors, what it does is before the entry of your mRNA into the site, they get all the things ready for the mRNA. What does they do is you have 40S subunit of your ribosome and you have tRNA. They make up this complex and they get attached. Once elongation eukaryotic initiation factors are released, when the mRNA and the ribosomal siRNA complex assemble. What does this mean? See, you have this is done by initiation factors. So you know the mRNA comes to this place and you have your 60S ribosome. Once this 60S ribosome subunit attaches to the mRNA, what has happened is all these initiation factors will get out from this side. Okay, so that you have over here, once all this done, you have mRNA along with your 40S subunit with tRNA and with 60S subunit. So this is the complex that you have after the process of all this initiation. And this assembly of your 60S ribosomal subunit to your mRNA requires GTP. Okay. Yeah. So this is about initiation. Now we'll go into elongation. What do you mean by elongation? See, you have. Sorry for using the term mRNA these many times because you need to use over here. This is mRNA and you have 40S. Let's have human model, not the cloverleaf we have discussed, and this is 60S subunit. Okay, this is all what you have before the elongation starts. So now amino acid tRNA binds to the A site. What do you mean by A site? A site is nothing but acceptor site. Okay, except for initiation methionine, because initiation methionine has already been there, which binds to the P site. Requires elongation factors and GTP. Already you require one GTP unit for initiation and another GTP for your elongation. Now this ribosome enzyme, what is this? Is 20S rRNA. This ribosome enzyme catalyzes the peptide bond formation, transfers the growing polypeptide to amino acid in A site. Okay, you have A site and you have T site, you have T site. This is about tRNA. Now we are discussing about ribosome. Do you understand the difference? It's all occurring in the ribosome. So in ribosome, wait, I'll show in the diagram. Yeah, in ribosome you have A site, you have P site. Okay, what does this A site means? A site will accept the incoming. So when a newly formed tRNA will come with amino acid, the A site will accept this and transfers this site to and transfers this amino acids to P site, and P site has 28S rRNA, which is ribosome, which catalyzes the formation of peptide bond. Now whole this sequence will move forward. Okay, once this move forward, and again new amino acid will be synthesized. See, so new amino acid will come. Ribosome moves along the left to right, along the mRNA. See, it is moving from left to right, and which forms the peptide chain. Okay, see, elongation, peptide chain. Once I'll recap all these steps. So first you have tRNA, newly formed tRNA, new tRNA, which comes attaches to your A site. Already you have one methionine carrying tRNA at your P site. So once this elongation process will initiate, this A site mRNA, A site containing amino acids will be transferred from P site to A site. Once this P site to A site transfer will not occur, what basically happening is, once this ribosome will move from the left to right, your A site becomes P site, and this site becomes A site. Are you getting the point? So your P site gets synthesize the new chain, peptide chain. Are you getting the point? See, let's say this is a P site, this is a site, and you have a methionine carrying a tRNA over here, and you have another carrying histidine over here. Once the translation process is going on, when the elongation process is going on, as it shifts forward, what will happen is, let's say this is the new position, you have another codon over here, no? So this becomes your A site, and this old A site will become the P site. Okay, while this becoming the P site, this methionine will get transferred to over here, which forms the new chain, and another new tRNA will comes over here. Hope I have cleared, because this is a quite complex topic. Okay, ribosomal RNA catalyze the peptide from and transfers going peptide to polypeptide to amino acid in A site. Okay, first you have a clarity about what is P site, what is A site. See, A site, what does A site means? A site, P site already have your methionine tRNA. A site already have gotten the new tRNA. Once the ribosome has formed, moving on, the A site becomes P site. Whereas new P site, new A site will set in. Your A site will become P site. This P site have all your growing tRNA. Along with its chain. Okay, see, A site is for incoming, P site is for accommodating peptide, E site holds the empty tRNA as it exits. This is E site. Ribosome advances 3 nucleotides towards 3' end of mRNA, moving polypeptide tRNA to P site. Elongation factors are target for bacterial forms, diphtheria, Pseudomonas. So let's have some topic over here. You have some bacterial toxins which targets this protein translation. What are these? Decrease protein synthesis. Remember as decrease protein synthesis. Okay, decrease protein synthesis. So diphtheria and Pseudomonas. This will target your elongation factors. S is nothing but 60 S and Shigella like toxin of E. coli. So all this will target your protein synthesis and decrease the protein synthesis. Okay, clarified. Now you have a newly formed peptide chain over here, and you need to terminate. See, your P site have the newly formed chain, and you need to terminate the sequence. When does this terminate? When you have your UAG, you are gone, you are away, and then another one is you go away. When these codons are recognized by the release factors, then your translation will complete. See, elongation release factors recognize the stop codon. These are stop codons, and whole translation completed polypeptide is released from the ribosome. This requires GTP. Shine-Dalga sequence. This is important. Shine-Dalga sequence are present in prokaryotes. How do you remember this? Shine like a pro. Shine like a pro. Okay. Ribosomal binding site in prokaryotic mRNA. So these are the ribosomal binding site in prokaryotic mRNA recognized by 16S RNA in ribosomal subunit. Enable protein synthesis initiation by aligning ribosome with start codon so that the code is readily read correctly. So I'll explain this. Let's say this is all your ribosome. So this is your mRNA, and the thread that is coming is nothing but amino acids. What this mission does is it stitches all these amino acids to your mRNA. Okay, Have you understand this? Okay. Hope so. Post-transcriptional modifications, trimming, covalent alteration. So we have post-transcriptional modifications and post-translational modifications. In post-translational modification, we have trimming and covalent alteration. What does this mean? Removal of N or C terminal propeptides from zymogen to generate mature protein. What does this mean? You have trypsinogen and you have converted it into trypsin, and you have a insulin which is formed with C peptide. And once you remove the C peptide, and you have active form of insulin. This is also form of trimming. Covalent alteration is by phosphorylation, glycosylation, hydroxylation, methylation, acetylation, and ubiquination. See, this is important. Ubiquination is for protein degradation. Protein folding. This is an ATP dependent process. Okay. Now you have Chaperon proteins. These are intracellular protein involved in facilitating and maintaining protein folding. In yeast, heat shock proteins HSP60 are constitutively expressed but expression may increase with high temperatures, acidic pH, hypoxia to prevent protein for denaturation or misfolding. What does this Chaperon proteins are nothing but your intracellular proteins. What does this help in? They help in protein folding. Okay. So in yeast and in the yeast, these are nothing but heat shock proteins, and they are expressed as per the environment. So when they are required, they are expressed in more, and when they are not required, they are expressed in less. Okay, so we'll discuss this